Hi class, Dr. Jim here. In this lecture, we're going to look at the structure of the neuron. So we're going to really take a close look and see how the neuron works, how action potentials are generated, and really how they send them across the cells. And then finally look at the spot in between the neurons or the neuromuscular junction called synapses and see how the action potential, once it's generated in the nerve, how it's passed along the nerve and then meets the synapse and then triggers a chemical response or release that will then either excite or inhibit the response in the motor or the muscle tissue or other neurons as it goes along. And so we're going to take a look at how this is all generated, kind of look at how these messages are relayed. And a lot of times what I'll do is kind of mention when things don't work right. So, like in situations like MS, what happens and why the action potentials get screwed up or other things that happen there. We'll also take a brief look at some of the neurotransmitters that are out there and what they do. So, again, between the synapse. And then finally, I'll do a really quick look at the effects that different drugs do play on the nervous system. And so, a lot of us do things like caffeine, some of us smoke, some of us do some other types of drugs, and they do have an effect on the nervous system. And so, whether we're talking about mental illness or other effects, maybe even certain drugs like yeah, Benadryl and that, and what effect does it have on the neuron? These all play different roles in how we send and receive signals with the external environment. So let's take a look at how these wonderful things work and how our system works overall and generate signals for us to do everyday life as a human. So let's take a look at that. So the first thing we're going to look at is how do neurons work and how does their function affect information transfer. So again, we'll talk about the basic structure of the neuron, what types of neurons are located where, and what do they do, and look at those functions. We'll first then discuss the resting potentials. So what happens when the nerve is quiet, and so why does a certain signal trigger a response? And then we'll look at the action potential. And again, the action potential is essentially the response that we have in the nerve. And what you're going to see, it's really just an opening and closing of a lot of these gated channels. And so it's an electrical signal because we look at the charge difference from inside and outside the membrane. But really, it's a chemical that is secreted either in or out, and it works with action or active pumps and then also looking at diffusion. So again, we have to go to that good old active transport and passive transport and look at the difference and how that affects the neuron. And then finally, we'll look at the synapses and again talk about the differences in the synapses. Synapses work a little bit different than the action potential, but the action potential causes a release of a neurotransmitter, and then that causes the triggering either of another nerve or at the muscle, stimulates the muscle. And we're going to look at what muscles do in chapter 39, and we'll look at a few other things as we go along. But today we're just looking at the neuron itself and then kind of going from there and seeing how all this works. Okay, so let's take a look and see what a neuron actually is. And so the first thing we want to introduce is how animals respond. And so again, we've been looking at animals throughout the last couple of chapters and looking at their different systems. Well, the nervous system is no different. And what that allows for animals to do is respond and behave or react to their environment. And so really the next couple of chapters are going to look at one, how the neurons work, two, how the brain works, how the senses work, and then finally what happens with our muscles and how do we respond and the behaviors we get from that. And so that's really the next three chapters looking at all these different things. Now, again, neurons are nerve cells that transfer information with the body, and there's two types of signals to communicate, either the electrical signals, which are the long transmission, and that's again the generation of the positive and negative charge of the membrane potential, and then the chemical signals, which is what we see between the synapses. So with the release of the chemicals from one end of the synapse to the other end, which is either another neuron or the muscle in that sense. And so we'll look at that as well. And so this is one of the animals that responds, and what he'll do is he'll use this siphon to kind of see what is out there. So he'll use this as a sensing tool, and when he responds to something, he'll release his proboscis or his proboscis out of the siphon and snag a fish. And so we'll look at that here in just a second and how he does that. Now, with the neuron, the interpreting of signals in the nervous systems involves sorting out a complex set of paths and connections. And what we have are neurons that are associated in a ganglia, where you have multiple neurons or clusters of neurons. And here's one of ganglia in the spinal cord. And so you can see outside the spinal cord, we have these ganglia, which are essentially a collection of neuron heads or cell bodies that are here in the sense of the neurons. And again, they kind of sense what's going on and perceive signals and figure out what signals are the right signals to send out and look at those things. And then we talk about the brain, which is another very large, complex organization of neurons. Now, in the next chapter, in chapter 38, we're going to talk all about the brain. So this is really about the only spot that you'll see anything about the brain today and interpreting signals. We'll talk about the different aspects of the brain, the different spots in the brain, and what each spot of the brain actually does. But for today, we're just going to be concerned with how does the neuron work and how does it perceive signals going across the nervous system. Now, the neuron itself is a cell type that exemplifies the close fit and form of function that arises over the course of evolution. And with this, we have the dendrites, we have the cell body, and then we have the axon. And again, we're going to talk more about this as we go in throughout this chapter. But essentially, how neurons communicate with each other is they send a signal down the axon that gets perceived at the body or the dendrites themselves, and then that signal then gets transferred and then passed down the postsynaptic cell, which then can go to more terminals, which can either respond to other neurons or to muscles or other glands or other things to send out a signal. And so, again, this is what we're going to look at today: how this all happens. How does it get a signal? How does it send it down the course of the axon? And then finally, when it gets to the synapse, how does it trigger the next response? And so we'll look at that today and how that goes. Now, most of the organelles are in the cell body, and depending on the type of neuron you look at, the cell body can be in different spots. Typically, with the most neurons, the cell body is going to be right at the end where you have the dendrites, and these little looks like little roots are the dendrites that come off, and those are kind of the feelers. They're the ones that are responding to the environment. They're getting the stimulus or the signal. And now there can be other neurons that are or other axons that are attached to this, and we'll talk about that, where they have the synapse, and you can kind of see that down here where they may not even be attached to the dendrites themselves. So this guy can either signal by itself, getting a signal from the dendrite itself, or it can get a signal from this axon from the other neuron. And so that's what's going on here. Now, like I said, the signal comes in, or the stimulus comes into the dendrites, gets into the cell body, and then you have this longer dendrite, which is called an axon, and that's where the signal goes down. And it's kind of like the tail, or sometimes they call it the tail fiber of the neuron. And what that does is it sends the signal down to the axon to the either next neuron or to the sensory response or the muscle or the gland that it's going to respond to, and that would be the motor neuron. And we'll talk about that here in a second. But one of the important things is kind of this cone shape. This is called the axon hillock, and this is where the signal is going to begin. So we'll talk about resting potentials and then the action potential. And typically in a neuron, when it sends a signal, that signal is going to begin in the axon hillock and then proceed down the axon to the synapse. And so we'll look at that as we go along and talk about those here in a few minutes. Now, at the synapse, this is where the axon transmits the signal to the next spot. So it can either be a dendrite, it could be the cell body, or it could be a muscle or a gland or whatever else we're talking about, depending on the type of neuron that we're looking at. Now, at the synapse, what you have is that you're going to have the action potential come down, and that's going to trigger the neurotransmitter to be released. Now, it can either be transported out through a channel, or it could be like exocytosis and release all at once. And so we'll look at some of the different types that are out there and what they do. Typically on the postsynaptic receptor, you have a cell receptor that will bind the neurotransmitter. And again, we'll look at some of these things as we go on later on, of how these guys respond to a signal. And you can have both a positive and a negative signal that comes out of these things. So again, positive signal will cause the response to continue on. An inhibitory or negative signal will cause this, the system to shut down. And so we'll look at that as well and how those things happen as they go along. Now, when we look at the neuron structure and function, again, one of the cells that supports this is the glia cells. And the glia cells support the neurons. They help nourish the cells. They help the cells recover if they have any damage to them. They also help any and everything around the neuron. So it helps with the myelin sheath around the neuron. It helps do a lot of other things. And in our brains, we have more glia cells than neurons by anywhere from a 10 to 50 fold. And again, they help nourish those cells and they help support the nerve cells that are found in the brain and the rest of the body. And so we'll look at that as well. And so these glia cells are very important. And like I said, they help with the nourishment, they help with the myelin sheath, they help kind of take care of these neurons. They're kind of like the babysitters of the neurons. And so if you think it like that, or the housekeeper or the nanny of the neurons, so to kind of think of it that way. So they're a very important cell that has a lot of functions to help maintain the nervous system in general. Now, the nervous system processes the information in 3 stages. So first, it will get a sensory input. So in this case with the snail, it looks at the siphon, and this is kind of taking a feel of the water. It's like, what's going on out here? And then once it gets a sensory input, it will then integrate that inside its ganglia, if that's all it has, or small little brain, like in this case with the snail, it has a small little brain. It will interpret that signal saying, there's a fish nearby. Let's stick the little proboscis out and capture that fish. And what it will do is then send a signal to the motor neurons, which will then cause the proboscis to come out, stab that fish, use the neurotoxin that it does to shut it down, and harpoon and grab the prey. And so that's typically what happens in all animal cells. And so animals, what they will do is that they'll sense something out in their environment, or maybe internally as well. So again, in some larger animals, they can really do a good job of sensing their internal environment and respond to that. But in most situations, when we think of sensory, we think of outside the body. So what they'll do is they'll sense what's going on outside, they'll integrate it inside the inner neurons and the ganglia in the brain, and then they'll send a signal back through the motor neurons to do a response. And so that's typically how we think of how neurons are going to work and how the nervous system works in these situations. Now, like I've said before, we have different types of neurons. The first type is the sensory neurons, and they transmit information from external stimuli or internal conditions. Typically with the sensory neuron, now you remember seeing the picture of the neuron before, and I said sometimes they look a little bit different depending on what they look like. Now in this case, what you'll see is that the dendrites are here, and then the axon has this long tail, and then it separates out at the end into different types of things. Now remember, the axon is what sends the signal. What you'll see here is that the cell body is kind of in the middle. Now in the picture that we showed before, we showed normally the axon comes off the cell body, and that's where those things are. In a sensory neuron, the cell body is kind of in the middle. And again, I think that's more of an evolutionary type of thing. Not sure why they're in the middle there and that stuff. You'd probably have to talk to a neurobiologist to find out the real facts on that. But I'm sure it has something to do with the form and function of these things. Now, in an inner neuron, what you'll see is it's more closely resembles what a neuron does. But you can see an inner neuron has lots of different pathways in this axon. So it doesn't have it has a long axon, again, that's the red, but what you see is there's a lot of branches, and that's because it talks to a lot of different neurons and it can send signals to a lot of different things. So the inner neuron is very important for getting the signal and figuring out where it needs to go. So it's kind of like the shipping center of the neuron and saying, or of the nervous system, saying, okay, we're processing these different things that are coming into us. What do we need to send out? Where do we have to send this out to? Do we need to send out a quick motor response to pull their finger off the hot stove? Or do we need to interpret it? Do we need to send it to the brain? Where do we need to go? And so that's what the inner neurons do. Now, the motor neurons, again, look more like the typical neuron that we think of, where, again, we have a lot of these different dendrites coming in. We have the long axon tail that sends it out, and then that will be towards either the muscle or gland. So typically, it goes and it triggers either the muscle or gland. Sometimes they are connected to other neurons that are farther down the system, but most likely what you're going to have at the end of these synapses is either the muscle or a gland, which is going to cause a response. And so either you move that muscle or you secrete something or something else is going to go on there. And again, most of the time when we think of motor neurons, we think of motor or moving. And so that's going to cause the muscle to contract. And again, in chapter 38, we're going to look at how the motor neuron triggers the muscle to respond. Actually, it's in 39, chapter 39, when we look at how the muscles move and respond to the action potential that the motor neuron generates across the synapse as well. Okay, now we can break the nervous system down to two parts, and we're going to talk more about this in chapter 38 when we look at the brain versus the peripheral, so the sensory organs versus the central nervous system. And really what you can think of as the central nervous system is going to be the spinal cord and the brain, and that's really where you have a lot of the inner neurons. You may have a few sensory neurons coming in and that stuff. You may have some motor neurons coming out, but really the central nervous system is mostly the inner neurons to process and integrate those signals that are coming in from the sensory and sending out to the motor. And so that's what's going on there. The other system that we have in the nervous system is the peripheral nervous system, and those are all the neurons that go out. And so again, you either have the sensory neurons coming into the central nervous system, or you have the motor neurons going out. And so again, you think of the cranial nerves, the spinal nerves, the peripheral nerves. Those are going to be the ones outside of the central nervous system, coming off the spinal cord or the brain. And so that's where you see those. And again, what that contains is the sensory neurons and the motor neurons. And so the PNS neurons are bundled together to form the nerves, and the nerves are either the ones that get the signal in, and that's a sensory neuron, or send the signal out from the brain and spinal cord, which is the motor neuron. Now, how does this all work? So how do neurons actually work? And so essentially what you're thinking about, these are electrical signals that are sent down. So it's kind of like a wire in your house. And so your wire in your house has, again, two different wires on the inside. It has a positive and a negative, well, kind of a positive and a neutral charge in there. And then it has the insulation around it. And it's kind of what is going on in the nerve cell itself. And all of this is based on the action potentials or the potentials of the cell membrane. And so we're going to look at that. And so inside the cell is negatively charged relative to the outside of the cell, and that is due to the concentration gradients that you have. Now the difference in that energy is termed the membrane potential, and the resting potential is the neuron not sending out any signals, and so that's when the nerve is quiet. And when it's quiet, it's typically negative on the inside and positive on the outside. And again, it's due to the concentration of the different ions that are there. And it really works on the sodium-potassium pump that's going on here. And so we'll look at this, and again, the different pumps that are there, pumping it in and pumping it out. And so this book does a really nice job of really taking you through the difference between a resting potential and active potential and what actually causes a signal to go down the neuron. With the changes in the membrane, the potential acts as signals, transmitting and processing information. And so first what we're going to do is look at how a signal is produced inside the neuron, how it sends it down the axon, and then finally when we get to the postsynaptic cleft, we're going to look at the synapse from the neuron that's sending the signal to the neuron that's accepting the signal, and we'll look at that. And again, the active potential stimulates the neurotransmitter to be released at the end of the synapse to go and tickle the receptors on the muscle or the other neuron that it's receiving. And we'll talk how that all works here in a little while. Now, when we look at these things, the potassium and sodium play an essential role for forming the resting potential. And in most neurons, the concentration of potassium is higher inside the cell, while the concentration of the sodium is higher outside the cell. And because of that, and the amounts that are there, the sodium is higher outside, the potassium is higher inside. But even though they're both positive charged, you have less of the potassium inside than you do outside, along with these negatively charged proteins and other negatively charged ions on the inside that allow for this negative charge to generate on the outside of this, or in the inside of the cell, inside of the cytosol. Outside, because you have so much sodium out here, along with other protons and potassium out there, you generate a positive charge on the outside here. And a lot of this has to do with the different gated channels. And again, some channels are working passively because, again, you're working with a concentration gradient here between the two. And then you also have the sodium ion exchange pump that requires ATP to work. And so all these things are working together. These pumps use ATP to generate and create gradients across the potential. Because remember, in passive, it's moving with the gradient. And when you need ATP, you're doing active transport, and that's moving against the gradient. So you're pumping potassium out, or I'm sorry, bringing it back in because you have more in the inside here and it wants to go out. And sodium, you're pumping out, even though you have a higher concentration of sodium out, it wants to come in. And so it's kind of working with the system. And when it does that, it pumps these things out so that now you generate this negative charge on the inside and positive on the outside. And we're going to look at how we've noticed these things with the membrane potential. We actually stick probes into nerves and we can measure. And this is how they figured out what is positive, negative, and how these actually work by putting these little tiny probes into these nerves and seeing how they actually work. And so we'll talk more about that as we go along today. Now, it is due to the opening of the ion channels, and again, it's the difference between the sodium-potassium pump, which again is an active transport. These are all passive transport, these channels. So these guys, the orange and the brown, or the orange and the yellows, are all passive transport because they're going with the gradient. This purple guy is an active transport, so it requires ATP to work, and it's going against the grain and against the gradient. So we'll look at that as well. Now, as a resting neuron has many open potassium channels, allowing for the potassium to flow out, so it's generating a negative charge. The resulting buildup of negative charge within the neuron is the major source of the membrane potential. And so that's going to play a big role of why we can get these action potentials to work. You want a negative charge on the inside, positive charge on the outside. So this normal movement of these potassium ions really helps establish this charge. And we'll see this here in just a second. So we can actually demonstrate this in the lab by just creating essentially having an inner and outer chamber and separating these chambers out by these fake selectively permeable membranes. And so you can have these channels and essentially see the difference in the charge. So when you have an artificial membrane in the outer chamber, you have a positive charge out here because you're pumping the potassium out here and a very small amount of potassium chloride. And so the potassium chloride is lower out here, so this is going to be positive. It's going to be higher on the inside, so that's negative, along with the potassium here getting pumped out. And so you're generating a positive charge here and a negative charge here. And what you can see is the difference in voltage being about minus 90 millivolts. And you can figure this out by looking at the differences, and it's the 62 millivolts times the log of 5 millimolar on the outside over 140 on the inside gives you a negative charge of a minus 90 millivolts. And that's how they kind of calculate the negative charge and the resting potential that goes on there. And so really this tells you that potassium is diffusing down the gradient out to the outer chamber. So you're generating positive here. The chloride builds in the inner chamber, so that's generating the negative charge, and at equilibrium the gradients are balanced, and so with that you get a negative charge in this situation. Now the equilibrium potential of the membrane is based on this calculation, and so this is how they figure out what the charge is on the inside and outside based on the ion concentration. And so you always do the 62 millivolts. This is the standard number. And then what you have is the log of the ion outside over the ions on the inside. And so you can calculate what is the equilibrium potential of the ion in the membrane or between the membrane by using this formula. So the equilibrium potential for potassium is a minus 90 millivolts, and the resting potential of the actual neuron is between minus 60 and minus 80 because of the sodium coming in. Sodium is high on the outside, and it's going to be passively diffused on the inside using those channel proteins. And so it's not a complete minus 90, but the resting potential is anywhere from minus 60 to minus 80. And so that's going to be important for the resting potential, so we can generate an active action potential here in just a minute, and that will be a positive charge on the inside of the cell. So we'll look at that here in just a second. So in a resting neuron, the currents of potassium and sodium are equal and opposite, and the resting potentials across the membrane remain steady. So again, if you look at this, the sodium chloride is moving in, the sodium's moving in, while I'm sorry, moving in, where this is moving out. And so again, looking at the sodium potential with sodium versus chloride, you can see now on the inside you have a positive and you have a negative on the outside due to the concentrations. And so it's kind of the opposite of the opposite effect that we saw with the potassium in this case, but it's the chloride that's going to provide the negative charge in these situations. Now, again, we've seen this in a number of other places, and so this little slide here, the next couple of slides are just going to look at where we see these positive and negative charges and the action potentials again for these different areas. So one place that we see, again, the transport of sodium and chloride is with osmoregulation. And so pumping out sodium along with chloride to get the concentration, especially in fish that live in salt water. So water and salt is trying to get in, water's trying to diffuse out of the cell. And so in order to keep the water in, they keep pumping these sodium and chloride ions out so that they can keep the water inside the animal and not dehydrate too much. And so that's one of the things that can happen there. Another place is information processing in the neuron. And so we'll look at the difference between the sodium coming in and the sodium being blocked out. And so again, generating a positive charge on the membrane. And then looking at gas exchange, we mentioned this before. What causes the stomata to open is actually the introduction of the potassium in and the hydrogen ions out. And so that positive charge causes the stomata to open up in this case, along with the water coming in. And again, it's due to the concentration gradients that happen here. And then we also think of the positive charge to allow for the flagellum to spin. So in bacterium, they use the proton motive force that they generate when they're making ATP. That hydrogen proton actually spins the flagellum around. And so that's part of the reason why the flagellum spin is due to the proton motive force that's generated when protons are pumped out during the process of cell respiration. And so that's another place where you see gradients playing a big role. So how are action potentials generated? So we can measure the action potential by, again, using this system. And so you have this microelectrode attached to the neuron, and we can measure with the reference electrode what is going on inside the neuron. Now, changes in the membrane potential occur because the neurons contain gated ion channels that either open and close due to stimuli, and the voltage gated ion channel opens and closes in response to the shift of the voltage across the plasma membrane. So again, it's this opening and closing of these gated channels that allow for charges to come in or charges to leave. And we're going to see how this all generates a active charge that then sends it down the axon to respond to a sensory system that's out there, or again coming from the interneuron and sending the message to the motor neuron, which is now going to send the signal out to either the gland or muscle. Now, with the voltage gated ion channel, you have the ions on the outside. The ions are in this state, the gate is closed, so the ions can't move through. When you have a change in the membrane potential, it opens up the gated channel, which now allows for the ions to flow in and change the charge within inside the membrane. And so we're really going to see this with the sodium. The sodium is going to open up during the action potential, allow for the sodium to come in, which will change this from a negative charge out here to a positive charge, and this is going to become negative. That was once a positive charge out here. And so again, you're going to have a flipping of the charges when we see this response. Now, when the gated potassium channels open, potassium diffuses out and it makes the inside of the cell more negative. So again, you're leaving the potassium, which is a positive charge, letting it go out, and we call this a hyperpolarization. So with the resting potential, sometimes that charge can get even lower. So normally the resting potential, like we said, is anywhere from -60 to -80. So with a hyperpolarization, when you let more potassium out, so that gated channel is open, potassium's coming out, Now what happens is you get this thing called a hyperpolarization, which lowers the resting potential in this sense. So now it goes from a -60 to maybe a -80 or maybe even a -90 in this situation due to the stimulus. And again, the stimulus would be to open up the potassium channels to work its way out. Now, when we open other types of ion channels, that causes a depolarization, which is a reduction in the magnitude of the membrane potential. So you can see here, you have these depolarizations, which will now raise the potential up. And so now it's becoming more positive. So there may be stimuli that opens the sodium gates, but not enough to get over the threshold. Once it hits the threshold, then we'll see the action potential. But what it's doing is making the membrane on the inside more positive. So you can see that the number is going from a minus, let's say a -60, closer to a -50 in this case. And so now you're getting that depolarization where it's close, but not enough yet to stimulate an action potential. Now, if you have graded potentials, those graded potentials can change the polarization with the magnitude depending on the strength of the stimulus. So if it's a really weak stimulus, you're only going to get these little depolarizations in the membrane, but not enough to generate a big charge or an action potential in these situations. A lot of times graded potentials will decay with the distance because again, it's not enough to stimulate an action potential, which will stimulate a signal down the axon. And so that would be a graded potential. So this would be something where you have a tiny stimulus, maybe like a little light touch, and it doesn't do anything. Like you can barely feel that touch, and you're not sending the signal. And it's something where it might be, where you kind of feel like there's something there, but there really isn't. So it'd be a really light touch in this case where you might not feel anything, so it's not causing any response whatsoever. Now, once you do get a response that you actually feel, so a very strong touch, what you generate is an action potential. So what happens is that you get a massive change in the membrane voltage called the action potential because the depolarization is over the threshold. So it has to hit this threshold, which I think is like around a -54 or something like that. And so what happens is you get that depolarization, and now that causes an action potential, and this guy now generates this positive charge on the inside. So you can see that it goes from a negative charge on the inside of the nerve cell to now a positive charge to almost about plus 30 in this case. And so these action potentials have a constant magnitude and transmit signals over a distance. So once you go past that threshold, that will now allow for the signal to proceed, and that signal will go, and again, typically with an action potential you achieve the same magnitude of height every time, so a +30 to +50 depending on how strong the signal is. That will generate an action potential which will send the signal down the nerve cell, and so now you have that, and that will travel over long distances. Now, when action potentials occur, whenever the depolarization goes over the membrane potential in a particular value called the threshold. And like I said, that's typically around -50. And so, the exact number is around 52, 54, something like that. And again, for each nerve, it's a little bit different. So that's why I say it's a little bit different. So just think around a -50. So if you get a depolarization that's above -50, so that would be, or I guess below -50, that's going to cause you to have an action potential. And so these are going to be the strong signal. So it's either all or none. There's no halfway or in between where you go whoop and just get a little bit. It's going to give a full rise, and again send that signal down the neuron. So this is a nice little video just to show you how the neurons work. So again, I probably didn't do a great job of explaining it. It's always nice to watch a video. I'm not going to show this here, but if you'd like to watch this, please do and get an idea of what this is. So when you go on the PowerPoint, or if you have that, watch it on the PowerPoint itself. So when you open it in on PowerPoint, you can actually watch this video and see. But I'm not going to run this today because for some reason it doesn't work very well on my computer and that stuff, but I'll let you do that and kind of see how this all works in this animation. All right, so let's just talk about the action potential and what happens with these gates. So this will kind of take it step by step of how an action potential is actually generated in these cases. And so you can follow along here with the nerve itself and getting the depolarization, hitting the action potential, and then what happens back to the resting potential. So the first thing that happens is that we need a resting potential, which is around -70 in this case, okay? And so in a resting potential, most of the voltage sodium channels are closed, so most of the sodium can't get in, okay? So it stays on the outside, generating a positive charge out here. And most of the potassium channels are also closed, in that you generate a negative charge. Now remember, potassium is going to be higher in the inside than outside, but you also have negative charge proteins along with chloride that's negative charge, and a number of other things that really generate a negative charge on the inside of the membrane. So think of that. And the proteins can't leave. And so that's why it stays more negative on the inside most of the time, because of all the negative charges you have inside. Proteins, ions, and other things that really generate that negative charge. And since all these guys are closed, even though they want to come across because it's a higher concentration out here, they can't come through because they are closed off. And so what you get is a negative resting potential in this situation. Now what happens is, now let's say you get a stimulus. So someone pokes you or touches you on the wrist or on your arm somewhere, or maybe you step on a nail or you touch something that's hot. What's going to happen is now you get this depolarization. So what the stimulus depolarizes the membrane. So now the sodium channels open first. It allows for the sodium to run in. And again, because of the passive transport, because sodium is high out here, it wants to come into the cell. So what you're doing now is you're causing a depolarization. So you're now getting more positive because more positive ions are coming in. And so if it hits that threshold, which is, like I said, just under -50, it's going to cause an active action potential to actually occur. So if the stimulus is strong enough, that depolarization is going to lead to an action potential. Now, during the rising phase, again, all the channels open up at once, and so that sodium comes in really quickly, and now you generate a positive side on the inside and a negative on the outside, because more of the sodium is rushing in. Remember, it's trying to balance out that sodium that's on the outside within the inside, so the sodium is going to rush in. That causes that positive charge now to occur on the inside. This becomes more negative. And so you get that really high action potential, which, like I said, is around +30 to +40. And then it reaches that height of magnitude. And then what happens is then you reach the falling phase, which is where now the sodium now closes back up, and now the potassium opens up and causes that passive transport out, because again, more potassium on the inside than the outside. So now what you're going to do is reverse the charge, because now you're losing the positive charge on the inside. That's going to make it more negative, and this is going to become more positive. And so now you see during the falling phase that it actually occurs. Now, because of this, you have what is called an undershoot, because these continue to open until you get a very negative charge in the inside. And so it actually goes below what the resting potential is, to about a -80 to a -90. And that's called the undershoot. And that is so that the cell can recover. So essentially, you're sending out more positive charge than what you really need to achieve a resting potential. But what it does is allows the signal to move forward. So that undershoot is important, so that the signal only travels in one direction. And I'll show you this in just a second. So that undershoot is really so that you don't get another response right away, and that the neuron can send that signal out throughout the axon. And so we'll look at that as it goes along. And then finally, what happens is once that undershoot is recovered, and we'll talk about that here in a second, it returns back to the resting potential, and now that neuron can hit the next spot. And so here you go. There's the threshold there, the number -55. Like I said, depending on the neurons, anywhere from -50 to -55, 52, 54. I've seen it in a number of books where they describe the average is about -54 in that case. But like I said, the undershoot will get it ready so that the neuron could respond again. But you need that time to recover in order for the neuron to send the signal down the axon. So again, during that refractory period, that's after the action potential, a second action potential cannot be initiated. So it allows for the cell to respond, again, send that message down, and allows it to recover so it gets back to that resting state. So that with this, the repolarization drops it down to probably, like I said, a -80 to -90. And that is due to result of the temporary inactivation of the sodium channel, so that it can't go out and back in. And so again, you go negative, really negative, and then the sodium channels are going to reopen and allow for some sodium to come back in, so that the charge will then be stabilized at the resting state, and you can send that down again. So typically where the site of the action potential is initiated is the axon hillock. That's where that, do you remember off the cell body? And the axon comes in and narrows down to the long tail. That's where it begins, so you can kind of see it here. This is where it's initiated, so you get the stimulus from the dendrite that tells the signal here and allows for the sodium to open up. So it's really the opening of that sodium gate that allows for the sodium to come in, causes a positive charge on the inside, and then what will happen is that the recovery period is going to shoot this guy down the axon so that you get there. And so it's the inactivated sodium channels behind the zone of polymerization that prevents the action potential from going backwards. So you want it to travel in one direction. So again, we'll see this, that the sodium opens up, then they close so that this can go down this way and not backwards to the dendrites again. Okay, so just showing you in general. So let's say we get an action potential here. The sodium channels open up. That causes a positive charge and a negative charge here. Then what happens is that due to the potassium coming back outside, reestablishes the signal, the sodium channels down, farther down will open up, allow for the positive charge in the inside. But due to this potassium opening up and the sodium closing, this allows for the repolarization of the neuron. And so again, you get that closing, so it's negative, and it's going to force it into this direction. And so it's a beautiful, elegant little system that allows it to respond. And remember, due to that resting part, what it does is it prevents the neuron from actually responding again until it is finally sent the signal all the way down. So it takes a little bit of time for the resting potential to reestablish itself so that the neuron can send the signal through the axon and then reestablish, and then it can get another action potential. But while it's in that resting stage, it cannot get another action potential. So you can't get these constant stimuli and allow this thing to keep going. It needs to recover before the nerve can send that signal again and again and again. So that's one of the things that has to happen. Now, one of the evolutionary adaptations of the axon structure is the production of these myelin sheaths, and these act like insulation. And what that does is it enables for fast conduction of action potentials, so it allows our nerves to respond even faster. If the nerve had to open and close all the sodium potassium channels all the way down, these responses would be very slow, and the animals would have to be very small. Once you add that myelin sheath, you now can bypass a lot of the axon, because what it does is it opens up here, creates an action potential, sends it through the myelin sheath, and then it will redo it at each one of these little nodes. And these are called the nodes of Ranvier. The nodes of Ranvier are essentially where the action potentials are generated through. And the Schwann cell, which covers the cell with the myelin sheath, is what produces that insulation so you can send that down. This myelin sheath, again, enables the fast conduction of the action potentials, and they are produced by the glia, the oligodendrocytes in the central nervous system, and the Schwann cells of the PNS. So again, these are, remember I talked about the glia and the Schwann cell, or I talked about the glia, I didn't talk about the Schwann, but both of these are important for maintaining the nervous system and making sure that the nerves are functioning properly and getting the signals across. And like I said, that insulation is really good because you're only going to generate those action potentials at the node and not in the myelin sheath itself. And we'll see that here in just a second. So again, action potentials are formed only at the nodes of Ranvier, the gaps in the myelin sheath where the voltage channels are found. And so what this allows is for a much faster conduction of the message because you only have these channels at these little spots. So it really allows to pass these down the node. And so you can have these very long nerve cells and pass a message very quickly. If you had sodium potassium channels all the way down, that message would travel much slower. And we do see evidence of this. So this is where I was going to talk about this, is that if you lose this myelin sheath, so in people with MS, what happens is an immune system actually attacks these cells and destroys the Schwann cell along with the myelin sheath. And when that happens, there's no sodium potassium pumps there, and so the action potential gets lost. And so even though you generate an action potential at the axon hillock, the message does it or gets lost in short circuits because there's no sodium potassium pumps in this place, and so the message kind of gets lost. So the action potential has nowhere to go. It triggers the first one, but then down the line it actually gets decreased and decreased because it kind of loses it. It's kind of like the telephone game where, again, if you don't have people in between, the message gets lost. And so that message gets lost, and so you get a short circuit. It's a lot like the insulation on your electrical wire. If you have a short circuit where the electrons can touch each other because the insulation is frayed, you get a short circuit because it crosses that line, and then you can get a potential fire. Now, in the case of MS, you don't get a fire with your nervous system, but you lose the signal and you lose that ability to respond and to move. And so that's the issue whenever we talk about MS. And so that's the idea. So with this myelinated, axons jump between the roads of Ranvier, and this is a process called saltatory conduction. It's a selective advantage for myelination is for space efficiency. So again, you can have long axons. So one, you don't need as many neurons, and two, you can have very long axons and send the message down very, very quickly. So that's the idea of having these things. And so it's a great evolutionary advantage. And so you see this on the higher level vertebrates because now they can have long neurons. The organisms actually can get bigger because they can send the messages much farther with this myelinated nervous system. Okay, now when we get to the end of the nerve, what happens? And so we have these things called synapses. And synapses are essentially the space between the nerve that's sending the signal to the nerve or the cell that's on the other side. Now, like I said, this could be a muscle, this could be a gland if we're talking about motor neurons. If this is a sensory neuron, this sensory neuron is coming in and it may be hooking up with either another sensory neuron that's sending the message, or it may be an interneuron, which is in the spinal cord or in the brain, and so it's getting the sense there. But there is this little gap between it, and that is called the synapse. And so once the action potential comes down, what it does is it triggers a chemical release that will allow for these neurotransmitters to be released and then trigger a response in the cell next to that. And so that's what we're seeing there. So that's what's going on in those situations. Okay, so again, in the presynaptic neuron, it synthesizes and passes the neurotransmitter through synaptic vesicles. So again, that neurotransmitter is being made at the end of the axon. Once the action potential comes down, it triggers this thing to become these synaptic vesicles to release as exocytosis, and that will bind to these voltage-gated channels or receptors on the postsynaptic membrane. And so that triggers a response on the next either neuron or muscle or gland to do what it's supposed to do. And again, the neurotransmitter diffuses across the synaptic cleft and is received by the postsynaptic cell. So you can kind of see message comes down, that triggers these vesicles to secrete. Those vesicles secrete out, and then they secrete this neurotransmitter to bind to the ligand channels on the next either neuron or muscle cell or gland, which will then cause a chemical response to occur here. If it's another neuron, you can see it's the potassium sodium, which triggers the action potential to go on throughout the next neuron. And the same type of thing does happen in a muscle cell, but we're going to look at that in chapter 39 and how that works. So this is another good little video. Like I said, I recommend watching this just to show you how the synapse actually works. So take a look at this. Again, and the action potential comes down, triggers a release of the synaptic vesicles, which releases the neurotransmitter, and then it binds to the receptor on the outside. So that's kind of the summary of this. Now we're going to look at how it actually works, what it triggers, and then some of the types of neurotransmitters that are actually out there. So with direct synaptic transmission involves the binding of the ligated neurotransmitters on the ligated channels. And so we just talked about this. So the action potential causes an increase of calcium to come into the neuron. So that's what happens here. That calcium causes a secretion of the neurotransmitter. The neurotransmitter is released, and then that will bind to ligated gated ion channels here, which will bind to the neurotransmitter, which will allow for sodium to come in and potassium come out. And again, this is an active transport in this case when it does that. And so we'll look at that, generating a postsynaptic potential in the cell. Now, there are two types of categories for postsynaptic potentials. You either have an excitatory, which means a positive response, so you get a depolarization of the signal. So this would be a positive response, or you get a negative response, which is a hyperpolarization, so it's an inhibitory signal. You can see the difference. The big difference is it sodium that's being allowed in, or is it the calcium? And so the sodium will trigger, the sodium gated channel will allow for sodium to come in, causing a positive charge on the inside. If it's an inhibitory, what will happen is chloride channels will open, and then you'll get even a more negative charge in this case. And you can see the potassium is coming out in these cases to generate a more negative charge or hyperpolarization. So that's the difference. So in a positive signal, sodium comes in, and a negative signal, chloride goes in. And so that's the idea in these situations. Very cool and very ingenious in how all these things work. So very, very neat that it uses the charges. Now, the cell body of one of the postsynaptic neurons may receive inputs from hundreds or thousands of synapses. And you can see here's the cell body of the postsynaptic neuron. That's pretty cool. And then you can see all the synaptic terminals of the presynaptic neurons that are coming into that one cell body. So you can see that it gets all these different signals. And so neurons have to figure out where is the signal coming from, and is it a positive or negative type of signal, and what is being done. And a lot of that has to do with what type of neurotransmitter is sent out. And so we'll take a look at that as well. Now, typically, one signal response is not enough to trigger an action potential in the postsynaptic. So typically, we think of a summation when you get multiple firings of these things. And so sometimes that's why you see axons that are multiple branch, so they can send out the name or send out the signal to trigger that postsynaptic neuron to get a summation of response in those cases. So like I said, the individual postsynaptic potentials can combine to produce a larger postsynaptic stimulation, cause a summation. So essentially what you have is you have one terminal branch, and so if it gets multiple signals coming down the same axon, what that will do is cause a summation. So you can see here that the threshold is not being hit in this first situation, so you're only getting one maybe action potential, and that's not enough to signal a message down that nerve. And so what you need is actually multiple signals coming in from that, either the same neuron or two neurons itself. And when it does that, it reaches a threshold, and now you get the action potential in there, and that's called a summation. So that's a summation when you have rapid succession or you get multiple ones working together. So again, here's the multiple ones where you get one coming from E1 and one coming from E2. That causes a summation, and now you see the action potential in the postsynaptic nerve. And so again, that can work together as well. So you can have one having multiple signals at one time, or a summation of the two. So again, depending on the type of neuron that we're talking about in these situations. Now, like I said, you can also get an inhibition. So even though you get an action potential from one, if you have an inhibition from another, it will quiet the summation and cause a hyperpolarization. So you can see that E1 is sent. It's not enough to do that, but it's also getting an inhibitor. So it actually causes a hyperpolarization. You get another signal from E1 and an inhibitor, and it's not enough to generate an action potential. So it's essentially shutting down the nerve. Now, sometimes you want this because maybe you don't want your nerves to respond. And so that will cause the sum effect so that you don't get that action potential and will never reach that threshold. And so that would be, again, the blockage of an excitatory response and an inhibitory response in this case, where you get the summation of the two and it shuts it down. Okay, and so in the synapses, the neurotransmitter to the receptor is called metabiotropic. And so a lot of times what that means is that not only do you have a gated channel, like in these cases where you have fast responses, but you may have some slow responses due to these metabotropic, it's hard to say, metabotropic receptors, which essentially is a slower response but a more or stronger and longer lasting response. And so what a metabotropic receptor is one that binds a neurotransmitter and does some type of metabolism that allows for the channels to open. So it's not a direct opening of a channel. So in this case, you can see here, this is a fast response where the neurotransmitter binds directly to a channel. In the metabotropic receptor, what happens is that you have a receptor that binds the neurotransmitter. That has to activate proteins, which will then go and open up an ion channel. In this case, you can see that this one even has a second messenger that produces and opens and closes ion channels along with other cell responses. This is what you typically see a lot of times in muscles and that. So this is a more slow, stronger response because you go through all these steps and almost a metabolitic or a catabolic response where you're activating proteins or enzymes, and then you turn on a second messenger. And so it's going to be a stronger response than a gated channel, and it's going to be longer lasting in this case. The problem is that it's a slow response because you have to go through all these steps first and to open the channel up to get the response and the action potential. So you need a stronger response, you need more time, but this ends up being a longer lasting and stronger response in this secondary messenger system. And that's kind of what I talked about here, is that in these guys, these are fast because, again, the neurotransmitter binds directly to the channel. In these cases, these are going to be slower because you have to go through a cellular process to open the channel and then eventually get a messenger. But these definitely last longer because these responses are slow, but you have all these other things going on, which takes time for them to go back to the normal condition. And so that's why it's a slower but stronger response. Okay. Now, the last thing we're going to look at today is the types of neurotransmitters. And like I said, typically there's a number of different neurotransmitters from the presynaptic cell that binds to the receptor. And a single neurotransmitter may have more than a dozen different receptors on the cell itself. And again, we're not going to get into too much about the receptors on the cells. We talked about the metabotropic versus the gated channels, and there are a couple like this. Acetylcholine has two different types. They have a gated channel and they have the metabotropic receptors, and it all depends on the type of signal that the cell wants. And so that cell wants a short, quick response. That's going to be a gated channel response. If it wants a short or long, or not a short, but a long-lasting response, then it's going to do more of a metabotropic response. And here's just a list of some of the structures of neurotransmitters. You can see they're very similar in structure. A lot of times they have these ring structures that would allow them to bind to the receptors. And again, a lot of these are positive in what they do. And then you also have some amino acids and some other proteins that will do some of the same things, but very similar structures in these neurotransmitters. And I'm not going to go through, I'm going to actually go through some of these here in a few minutes, but you can kind of see just what the signals are, and you can see some of the effects that they have. So if you want to pause the video and kind of take a look and see and that stuff, I'll zoom in for you so you can look at it real quick and then read what they do, the positive and negative responses. But we're going to go through some of these as we go along here in a few minutes. Now, like I said, the first one and the most common one is probably acetylcholine. This is for vital functions involving muscle stimulation. Typically, your muscles are associated or stimulated by acetylcholine and acetylcholine release. Memory formation in the brain is using acetylcholine, and learning is also responsible for acetylcholine. And there are two major classes of acetylcholine receptors, one that's ligand gated, so again, the gated channel, and then one is metabotropic. And so you have one of each. Again, the gated is going to be a quick response, very fast. So again, you need your muscles to respond, like getting your finger off the hot stove. That would be probably a gated channel because you want to move fast and say, here's the signal, get it off. Or if you want something that's more long lasting, maybe you're going to exert yourself and lift something up, so you need some contraction in the muscle. That's going to be more of a metabotropic type of response, where you want more of a long lasting contraction. So maybe you're holding something or doing something to lift something up, and that's going to be more of a metabotropic type of response in those cases. Here's acetylcholine, and again, I showed you what it looked like in the structures, and this is one of the major neurotransmitters that we have. Now, the best understood function is the ligand channel and vertebrate neuromuscular junction, and we're going to talk more about this in chapter 39, so I won't go into too much detail here, but essentially this is what we normally use as a synapse. And so what happens is the calcium comes into the cell. Once the nerve impulse comes in, it triggers the calcium to come in. That causes a release of these synaptic vesicles that are made in this area, so you have a lot of endoplasmic reticulum in this area that allows for these synaptic vesicles to be created. They go in and then they release this wonderful neurotransmitter to bind to these gated channels, which allows for sodium to come in and cause a response. And so we're going to look at this and what happens with the muscle, but you can see how this works. And so you find this receptor, and this is typically an excitatory response generated when the ion channel opens, and this is found elsewhere, both in the peripheral and central nervous system. So very common type of response that we see here with acetylcholine. Now, acetylcholine can also have, well, it's not a negative effect, but sometimes we see where acetylcholine can be affected. Now, in one situation, what happens is that you have acetylcholine. When it gets released, you have this acetylcholine choline esterase, which actually breaks down acetylcholine so that you don't get this overwhelming response. However, what happens, and so that's the inhibitory response, this little Paxon muscle or thing, which allows your muscles to relax. So in the case of sarin gas, what sarin gas does is it actually blocks this Pacman from eating up acetylcholine. So you get this overwhelming response, which causes this constant contraction. This is the same thing that happens in tetanus. And what happens is you get this contraction that will not be released because you get so much acetylcholine in your muscles and they can't stop contracting. And so we call that lockjaw, or we call that basically contraction, which leads to death due to the constant contraction, or tetanus. And so that's what goes on. You get tetanus when your muscles contract and you can't release that muscle. And so because the acetylcholine is just going over and over and triggering that muscle to keep releasing, because the acetylcholine keeps getting released, and there's nothing to eat up that acetylcholine to prevent the muscle from relaxing. And so that's the problem there. Now, there are some muscle relaxers that act like acetylcholineases, which will allow for acetylcholine to be eaten, so then the muscle can relax. And so a lot of times if you have, maybe you pull a muscle in the back and it's contracted really tight and it doesn't work, you may get prescribed a muscle relaxer. Well, what that will do a lot of times is it allows the muscle to relax. So essentially it prevents acetylcholine either from being released or being eaten up. It gets eaten up so that you now get this relaxation of the muscle, and then you feel better and you can kind of move around again when you take it. Now, like I said, acetylcholine is more than 100 known neurotransmitters, so there's a lot of other ones that are out there. But probably the most common type, like I said before, was that the most typical one you see with muscle cells is acetylcholine, and so that's the one that you normally see in this situation. Now, some other types of neurotransmitters include amino acids, including glutamate, which is used at the neuromuscular junction. Glycine acts as an inhibitory that lies outside the brain. Gamma aminobutyric acid, or GABA, is a neurotransmitter at most inhibitory synapses in the brain. So we talk about either inhibiting neurons or stimulating. So again, two of these kind of inhibit, where this other one, glutamate, stimulates at the neuromuscular junction as well. Now, some other chemicals, including amines, include norepinephrine, which is chemically similar to epinephrine, dopamine, and serotonin. These are active in the central and peripheral nervous system. And these biogenic amines have central role in a number of nervous system disorders and treatments. And so a lot of times people that have mental illness or other depression and other things are not making enough of these things. And so if they're not making enough, what we can do is give these drugs as a way to stimulate. Or sometimes the opposite effect is happening, where you're making too much of these things and you're being overwhelmed and you need drugs to slow these things down. And this is, again, we're going to talk about this here in a minute, but this is what happens when you take a lot of these different drugs, either pharmaceutical or recreational drugs. What they do is they mimic or inhibit or overstimulate these types of drugs, or these neurotransmitters, which cause different effects in the brain. And we'll talk about that here in just a second. So that's one of the things. But I just wanted to point these out. These are very important, especially in the brain and the brain chemistry that goes on. And so a lot of times people with mental illness are not making enough of these things. So they need antidepressants, which will actually help stimulate or mimic these types of these types of neurotransmitters to help them feel better. And so that's what's going on there. Now, there are some neuropeptides, which are short amino acids that also function as neurotransmitters. This includes endorphins. And again, you hear about endorphins a lot of times, which are the perception of pain. Sometimes we release those when we have feelings of happiness and joy, release those endorphins, or we work out and we feel good about ourselves. You get those endorphins, which really kind of tickle the neurons and, again, give those excitatory effects and make you feel better. And so the endorphins can do that. And again, one of the things that we see is a lot of times that these pharmaceutical drugs are these opiates. And so people, to cope with pain, take these opiates to block the pain receptors. But unfortunately, they abuse these things because they perceive pain more and their body can't work without these things. And so that's one of the issues. And so That's the problem with opiates, especially when they're severe. People can get easily addicted because their body recognizes it and says, that makes me feel good. And so I need more of it and crave that. And so that's part of the addiction process in those situations. Now, there's some other ones, some other chemicals like nitric oxide and carbon monoxide that are also regulators of the peripheral nervous system, but they're not neurotransmitters stored in vesicles, but they're synthesized as needed. Now, nitric oxide, we've talked about this before as a way to stimulate vasodilation in these situations. So we talked about this. I mentioned this. This is Viagra and that, and also in stimulating blood vessels in the heart. So you do see this a lot of times in the periphery, especially with the neurotransmitters, to stimulate dilation of the smooth muscle in our blood vessels. So that's typically where you see that. Carbon monoxide is nasty, and so that actually shuts down the nervous system. And so what happens is people that breathe in too much carbon monoxide will get obviously pain, but they'll also, what it does is it shuts down systems. And so we hear about carbon monoxide poisoning, especially now. So this is always the reminder to get your furnace checked and that have carbon monoxide sensors in your house because it's odorless, it's tasteless, and if your body brings that in, it can cause paralysis. And so you don't want that. So it shuts down the nervous system. So that's a nasty, nasty guy. Nitric oxide is the opposite. It stimulates the nervous system. Carbon monoxide actually shuts it down and it leads to paralysis and other things. So nasty guy versus pretty good guy where it stimulates the nervous system. Now, I did want to mention just a few quick minutes on the drugs and drug effects. There's a really good site out there. I should have put the link up here on the thing, but if you ask for it in the lab, maybe I'll bring this up in the lab when we do this. But it's called the Drug Mouse, and it's actually from the University of Utah, which is kind of the one place you wouldn't think that would have the drug mouse because you think of Utah and that being the holy place and that stuff, and people don't do any of those things. Well, at the University of Utah, they've actually studied and they do a really cool kind of this thing called drug mouse. And what they do is they put this little mouse in a container and they show you what actually happens to the poor little mouse. And it's all cartoon. It's all cartoon and that stuff. But it's really cool because it shows you the effects that drugs have on the nervous system. So they talk about cocaine, they talk about opiates, they talk about marijuana, they talk about even caffeine and that stuff and smoking, what those things do. And it's a really cool feedback kind of thing where you can put the mouse, you grab the little mouse, you put them in the box, and then you give them the drugs and that stuff. And it shows what it does to the nervous system, which is really kind of cool. So I'll bring that up in class and give you the link so that you can actually see that and use that and play with it a little bit because I think it's a lot of fun and you can actually see this. So, like I said, drugs that we take, a lot of these things we do to ourselves, but there are other things out there and they can block either the change in the neurotransmission or have an effect on the release or availability. And you can see what a lot of these things do. And so, a lot of these are drugs that we take, and what they do is they either increase impulses or slow things down. And so, that's the issue with these neurotransmitters and what they can do. And remember, some of these are inhibitory. So it may, even though it says an increase in neurotransmitter release, it could be in the inhibitory role. So that's what you see. So things like nicotine, alcohol, and opiates, they have both positive and negative, but they always increase the neurotransmitter release. Some of these, like the opiates and nicotine, are more addictive, and the reason for this is because they make you feel good. Alcohol a lot of times is an inhibitory and it makes you lose sensation. And so that's what happens if you drink too much. A lot of times you get kind of spaced out in that. And the reason for that is it kind of slows down the nervous system in that case. Amphetamines and methamphetamines, they speed it up. So this is what essentially is speed. This is what people would take. This is what's in meth. And so you hear about meth and methamphetamines. Why they take this drug is that makes them feel good. It makes them hyper, makes them feel like they're going to bounce through walls. And you probably have had this before if you've ever had cold medicine. And again, you may feel like you're going to bounce through the roof. A lot of these decongestants and that stuff that you now have to get behind the counter is because people would abuse them. They take a lot of these things and it makes you feel really jumpy and wants to basically run through a wall. That's because you get this increase in neurotransmitter release with those. And so that's what people feel and they bounce through. And that's why one of the reasons why, because people abuse this with the methamphetamines making these drugs, is why you now have to give your driver's license and basically a kidney in order to get any of these drugs that are behind the counter. Now, nicotine, like I said, can cause a response in neurotransmitter. It is highly addictive as well. And so obviously that's part of the cigarettes. cocaine and the amphetamine as well can block reuptake, and so again allowing for more neurotransmitter in the cleft, so again you get the stimulation effect. And so this is why a lot of times these become addictive, is because of what happens is that you get this overstimulation of the neurons, and then when the neurons don't get it, they say we want more, and so we need this. Again, there's some that are less neurotransmitter release, but we really don't have any drugs. And then caffeine and LSD. And I like this because most people don't realize about caffeine. They need their caffeine for their drug. But what it does is it can block receptors with another molecule. And so there may not be a change in the neurotransmitter release, but it cannot bind to the receptor or postsynaptic neuron. So again, a lot of times caffeine is one of those things that actually makes us feel awake. And what it does is it prevents the inhibitory neurons from actually responding, so you feel more awake in those situations. And so caffeine can be abused too, and a lot of people are addicted not even knowing it. So if you're one that needs about 5 glasses of coffee or cups of coffee a day, or one that needs to drink a 20-ounce soda every 5 minutes, you may be addicted to caffeine. You may want to slow yourself down, because that also has effects on your adrenal glands and stimulating adrenal hormones and other things in your body, as well as some other things. And so it can also cause weight increases in situations when you drink a lot of caffeine in that too. And so because the body's trying to respond to this overstimulation in the nervous system. LSD is acid, or another name of acid, and again that causes a hallucinogenic effect on the brain and makes you see things in that. And again, in that drug mouse, it really shows you what it does, and does a really good job with that. So like I said, I'll show you this in class, but I'll also give you the link in class too. So if you ever want to go and play with that and just see, whether or not you, like I said, I'm not going to say take drugs or not take drugs and that stuff. Well, you shouldn't take drugs, but it really gives you an idea of what would happen if you do take these drugs, and just show you maybe this is not such a good idea, and why people get addicted to those things. A lot of times because it enhances the reward center, which is dopamine, and that dopamine response, people crave that response because it makes you feel good, and that's why you get addicted to a lot of these different drugs. So again, that's what happens in those situations. So just touching the surface here, but like I said, when we do the drug mouse, it really will kind of show you in a cartoony kind of way of what really happens in these situations. Okay, so we made it to the end. So hopefully I gave you a good enough picture of how the neurons work in organization and how they do their thing. And so again, we have the central and peripheral nervous system. Typically, you get the sensory from the peripheral, goes to the inner neuron, which interprets the message, sends it to the brain or the other ganglia, and then that sends out a message to the motor neuron. We'll talk about reflexes and all that in the next thing with senses and all those good things. But again, we'll look at all that stuff as we work on. Today was more about just how does the neuron operate. Again, with the ion pumps, the ion channels establishing the resting potential of the neuron. So sodium, potassium, and chloride all play a role in establishing that resting potential. Remember, that means positive on the outside of the membrane, negative on the inside. And that also the negative proteins on the inside that can't leave also generate that negative charge. Action potentials are the signals conducted by the axons. So again, when they get a stimulus that causes the sodium channels to open, causing a change in that membrane potential. So now the outside becomes negative, the inside becomes positive, and that's going to send that signal down. You have that resting spot, that repolarization, which again changes that signal back to the negative charge in the inside, but it takes time for that active potential to come back, and so you need a little bit of rest before you can send out a signal. And because of the way the pumps work, it only sends it in one direction down the axon. And then when we looked at it, the neurons communicate with other cells at the synapses, and this is not the action potential causes the stimulation of the neurotransmitter to be released. And depending on the type of neurotransmitter, you can have excitatory neuron or excitatory synapses, or you can have inhibitory depending on the chemicals that are released. So you can have either excited and stimulate another action potential, or inhibit an action potential from actually occurring. And we looked at some of the different types of neurotransmitters out there, the types of responses. The quick response is the gated channel, where the metabotropic response is a slower but more long-lasting type of response. And like I said, quick response, hey, move that off the stove, dummy, because that's hot. or working out and building the muscles and contracting and holding something up, you need a longer response there. It's just kind of showing you those different things. So if you have any questions, please feel free to ask. Send me an email. Send me, ask me in class and that stuff. We'll definitely go over it again. I'll show you that drug mouse and that stuff, and that will be fun in the lab. So if you have any questions till then, please let me know. I thank you for watching, and I'll see you next time.